So the last video we left off after we discussed the leading strand being synthesized continuously in the 5' to 3' direction. So that was continuous synthesizing by DNA polymerase 5' to 3'. The lagging strand is being synthesized discontinuously. It lags behind in terms of where it's being produced. So the reason that we have to do that is because DNA polymerase will only work by synthesizing in the 5' and 3' direction. You can only place your DNA nucleotides down with the phosphate group on the 5' carbon and attach that 5' to the 3' hydroxyl group on the deoxyribose sugar. So the 5' to the 3' end. That's the only way you can synthesize that DNA, efficiently. So, in order to do that, we have to synthesize starting from our opening here. We need a new enzyme. We need an enzyme called primase, one that we haven't discussed with the leading strand, even though that is also working on the leading strand. But unlike with the lagging strand, the leading strand you only need primase once, the first time. And with the lagging strand, you need primase every time you start a new Okazaki fragment. Now, Okazaki fragments, by the way, are these short segments of DNA nucleotides attached together, and they're named after the team of researchers, the leader of the team of researchers, anyway, that first described and studied this, or first described this, anyway. Now, this RNA primer is going to lay down a few bases of RNA that are going to complementarily bind to the DNA. And then it's going to, how's the word I'm going to use? Allow the DNA polymerase enzyme to attach to that primer double strand and start that synthesis of 5' phosphate to the 3' of the RNA. So it has to prime it, or it won't start forming it at all. So when an Okazaki fragment starts, we lay down a primer at the opening of this replication fork. And then we put down a DNA polymerase, and then we synthesize until we hit the previous Okazaki fragment. And then, but while that's been happening, this replication fork has been opening up even more, opening up new bare DNA, allowing RNA primer, that's bare, B-A-R-E, so that the new primase has to set down and create a new primer right around there, and then a new DNA polymerase has to start a new Okazaki fragment. So you are building this DNA strand discontinuously. You're making multiple Okazaki fragments in short 200 base pair or so bursts, using a different DNA polymerase than you would for the leading strand, but it still does the same job. Now, like I said, DNA polymerase for the leading strand still does require a primer, but because the leading strand is synthesized continuously, you only need to prime that DNA strand once, the first time. Now, how are we going to get these Okazaki fragments to piece together? Because you've discontinuously synthesized this strand. So now you have your other strand of DNA, but it's all up in pieces. It's a jumbled mess. So you need another enzyme called DNA ligase. DNA ligase is going to ligate, which is tie in Latin, I believe. It's going to tie up these Okazaki fragments together, and they're going to fuse them into a single continuous strand. And that, in a nutshell, is how the replication fork is synthesizing both, is how you're synthesizing both DNA strands, the leading and the lagging strand, at the same time at the replication fork as it expands. Now, keep in mind, we had bubbles before. The replication fork is expanding in both directions at the same time. So this replication fork is only one side of that replication bubble. we need to flip this upside down and mirror reverse it on the other side, and then we will have something akin to what we're experiencing with a replication bubble. Now, here's a list of our terms, just in case, just as a recap. Our leading strand is going to be the parent DNA strand that runs in our 3' to 5' direction, and it can be replicated continuously by DNA polymerase 5' to 3'. Complementary, anti-parallel, double-stranded. The lagging strand is the parent DNA strand that runs 5' to 3, and you have to replicate it 5' to 3 in short spurts, so that the overall effect is replicating pieces in a 3 to 5 direction. So you replicate 5 to 3. You replicate 5 to 3 in short spurts, but you're doing 3 to 5 overall. Now, Okazaki fragments are those short spurts that we talked about for the lagging strand. They are formed in 5' to 3' direction as always. Now, as soon as you run out of space, as soon as you run to the origin point of the previous Okazaki fragment, you stop synthesizing the next part of you stop synthesizing that fragment. An Rna primer is a short segment of complementary RNA on the DNA strand. It placed on that DNA parent strand and it acts as a starting point, like the metal starter for a zip-up jacket. So this happens for both the leading and lagging strands, but more often for the lagging strand. Now, activated nucleotides. Now, I think I've discussed this when we talked about, in Unit 1, nucleotides. An activated nucleotide is a nucleotide triphosphate. It's got three phosphate groups instead of just the one. In order to produce your double strand or your single strand, in order to replicate that strand, in order to make sure that that phosphodiester bond is going to actually form, you require two additional phosphodiester bonds, two additional phosphate groups with their anhydride bonds to be broken to obtain the energy required to form that linkage. So an activated nucleotide, when we're talking about DNA, is going to be a DNTP as far as I was trained. That's a deoxyribonucleotide triphosphate. So an NTP is just ATP, TTP, GTP, and CTP, so DNTP. An NTP is just a nucleotide triphosphate for RNA, for example, so ATP, UTP, GTP, and CTP. Now, the proteins, DNA helicase, that is a protein that unwinds and separates the parent strands of DNA. And DNA polymerase is a protein that synthesizes DNA continuously in the 5' to 3' direction. And DNA ligase is a protein that joins the 5' and 3' ends of the DNA strands that were made by different DNA polymerase enzymes. So the Okazaki fragment ends, or the leading strand end when it runs into the new replication fork bubble, once those bubbles meet each other. Primase is a protein that lays down an RNA primer so that the DNA polymerase can actually attach and the replication can really get started. Now, don't ban the video, but I'm going to throw... actually, pause the video.